So the second part that we're going to go through is chromosome structure and specific epigenetic marks. So in this lecture and the subsequent number of lectures, we'll go through how epigenetic marks contribute to the ends and the centre of the chromosome that I mentioned already, and chromosome maintenance, and also how these specific epigenetic marks that we've mentioned, these particular punctuation marks that are laid down in the genome, what they mean at the molecular level. So we know that DNA is not just naked by itself in the nucleus. Instead, we know that DNA is wrapped around histone proteins, and together the DNA and histones make up the chromatin in the nucleus. The reason this packaging has to occur is that we have more than 2 meters of DNA within our nucleus. And so that 2 meters of DNA must be compressed down into a 10 micrometer diameter of nucleus, so a very small space. So the only way that you can think that this logically occurs is some ordered structure. If it was unordered, it'd be like a bad ball of string in your nucleus that was never really behaving as you'd want it to be. So this packaging means that although it means that it's much more ordered, it also creates its own problems. So we need to access the DNA at several times. First of all, during transcription, which we've talked about. So we need the RNA polymerase and the transcription factors to be able to get to the DNA sequence itself, but also during DNA replication or DNA repair. So at all of these times, you need to be able to access individual bases within the DNA. So the tightness of how tightly packed the chromatin is, how tightly packed this DNA and histones are, relates to the accessibility for these transcription factors and the RNA polymerase. So the general rule to think about is that if the DNA is tightly packed around the histone proteins, so the chromatin is very tightly packed, then it's inaccessible to the RNA polymerase and the transcription factors, and so it's less likely to be active or less likely to be expressed. And the reverse is also true. If it's loosely packaged and quite open, then of course the RNA polymerase and the transcription factors are much more likely to be able to get in and access those bases that they need to read. And so it's more likely to be active. If at this point you think, I need to go back and remind myself of what DNA replication is or what transcription or translation is, here are some links that you'll also find on the website. And these are for animations. So fairly short, a few minute animations that are showing the process, and so they're a very good little refresher that you could go and have a look at. So let's think again about these DNA and histones. Well, the smallest unit of the DNA plus histones is known as the nucleosome. So in the nucleosome, we have the DNA shown here wrapped 1 3/4 times around a histone octamer, so meaning 8 units of histones. These 8 units are 2 each of histones H2A, H2B, H3 and H4. So together, 2 each of these proteins make up the 8 histone molecules that are found within the centre of the nucleosome, acting like a bit of a spool for the DNA. And then on the outside, we have histone H1, which locks together that spool and holds the DNA in place. So the reason that this interaction can occur between the histones and the DNA is because histones are rich in positively charged amino acids, particularly lysine and arginine. So this means that overall histones are positively charged, and yet DNA overall is negatively charged, and so there's an electrostatic interaction between the two. The N-terminal tails of the histones, that is, we know that proteins are always N to C, have an N-terminus and a C-terminus. The N-terminal tails tend to actually protrude out of the nucleosome. And it's these protrusions out of the nucleosome that are important, and we'll come back to later when we think about epigenetic marks, particular epigenetic marks. So now I'm going to play you a short animation that's been made by WeHi TV, where I work. And you'll be able to see that these tails protrude out from the nucleosome in this animation. So here you can see the nucleosome, and it's got around it the DNA, and you can see each of the bases in the DNA. So there's purple DNA wrapping around the histone octamer, and you can see these long, pale grey, pale blue tails of the histones protruding out. Now each of those histones are coloured a different colour, so you can see that there are 8 different molecules that are found within this central spool of the histone, of the nucleosome. So a different way to look at this is using an electron micrograph, so a very high power microscope. And so here, this is an image from an electron microscope called an electron micrograph. And the black arrowhead shown here is pointing to a single nucleosome. And then this white arrowhead shown here is showing that there's a line, and this is the DNA that's between each of these nucleosomes. So these are nucleosomes with DNA in between them. And so these nucleosome plus linker DNA is known as beads on a string. And so while you can see here, and this is what actually happens in the nucleus, in parts of the nucleus. And so this is still, even with those histone proteins, is still a relatively open structure. There's a lot of this electron micrograph that's shown that does not contain any DNA or any nucleosomes at all. Another way to look at this is in a short movie, which I'll show you of the same sort that we saw earlier. Okay, so here you can see again the purple DNA wrapped around the histones, making up the nucleosomes, but it's in a beads and a string structure now. And you can also see the yellow sort of little fuzzy blobs bobbing around, but to the other molecules in the nucleus, and they're undergoing Brownian motion. So I'll show you again. You can see there's quite a lot of motion going on, and that's because this is still a relatively open chromatin structure. And so there's a lot of accessibility, even with these histones there, for the RNA polymerase or the transcription factors to come in and be able to bind to DNA as would be required for active chromatin. In the next lecture, we'll go into how extra chromatin packaging, how the chromosome is condensed further.